Good morning. It's quite loud this morning, excuse me. Sort of shocked myself too. Let's continue with our derivatives of the neural tube. And we were talking about the mesencephalon, but we didn't quite complete it. So I'd like to return to it. And we drew a section through the mesencephalon. We showed the aqueduct was here. This is our central canal as it looks. Boy, this is loud. Is that loud for you too? Or is right? It keeps you awake? It's what? It's okay? All right, thanks. Aqueduct. And then we had these could be the corpora quadrigemina either. I just repeat it, repeat it, repeat it. And sometimes that's referred to as the tectum. Tectum means roof. And you'll see that in the literature too, so I thought this morning I'd put it in. Tectum is the same as a roof here. Then we had the cerebral peduncles down on the ventral surface. And we put in the substantia nigra, which would be just dorsal to the peduncles. This would be the substantia nigra. And we said it gets its name, black substance, from the pigment melanin. Pigment melanin. And that the fibers were rich in dopamine. Dopamine was their transmitter. And the substantia nigra projects then up to the basal ganglia. We'll see the basal ganglia today. We'll develop it. Basal ganglia, which modifies motor behavior. Does many things, but just for the moment we need this. Modifies motor, we'll just say motor action. So that if we get some that knocks out our substantia nigra, so lose cells in substantia nigra, what disease did we say you got? Everybody knows that one. It's such a clear-cut one. Parkinson's. And the tremor for Parkinson's is a tremor at rest. And I give you that one because there's another part of the brain that gets destroyed, and it's a tremor when you intend to do something. So you be sure when you look at your neurosurgeon whether he's trembling at rest, because then you know he'll go away when he starts to do his surgery. But the intention tremor can be sitting there quietly, but when he picks up the scalpel, then he does that. So you have to be careful. So let's move on up then. There are lots more things in our... One other thing I do want to say, though, because I want to be consistent here. We've been giving the cranial nerves with each one of these areas as we move up the brainstem. So the cranial nerves with the mesencephalon will be 3 and 4. And the ventricle then was our aqueduct. So to see how they change as we go up. So our next area will be the diencephalon as we move up from the mesencephalon. our diencephalon. And we might as well give the cranial nerve associated with it, since we're just talking about it. What cranial nerve is related to the diencephalon? Well, we've moved all the way up. We've only got 2 more to go. We've gone from 12 up to 3. So where are we with diencephalon? 2. And what is the second cranial nerve? Does anybody know? It's the optic nerve. Very important. Yours is definitely being used right now. And the ventricle in the diencephalon then is going to be what ventricle? We've had 4th, we've had aqueduct, we're moving up. What's next? 3rd, right? So just so you can figure these things out for yourself, so it's not just memorizing, you can see where you are. So we're going to have a 3rd ventricle. And the structures that we'll be dealing with then will be the... I'm going to take this off. I trust you have it. The thalamus and epithalamus and hypothalamus. But I first want to make one point, because it gives a landmark that's used when you're using radiology and finding out where your sections are coming from. So I'm going to make our old neural tube that we had. This is our neural tube. And the most anterior part of the neural tube is called the lamina terminalis. Lamina, which just means layer, terminalis. Because we want to use this landmark when we put in our diencephalic structures. So this is going to be our lamina terminalis for a lateral view of our diencephalon. This is our lamina terminalis. But important structure for a landmark when you're reading scans. So from it, we're going to come out and form the structures of the diencephalon. And what we've put here, we'll have 3 structures. We'll have the epithalamus. Epi means upon, so it means upon the thalamus. So the epithalamus is just going to be some structures up on top here. But in addition, the pineal gland back here. So our epithalamus will be this, and this will be our pineal gland. It's the only structure of the epithalamus that I'm going to give you. It is what? It's a neuroendocrine gland. Neuroendocrine. It plays a role in establishing and maintaining your biological rhythm, your biological clock, if you like, biological clock. Don't you find it strange that after you've gotten up at 6 o'clock in the morning for several months and had your alarm, sometimes your alarm doesn't go off, but you get up anyhow? Your pineal is playing a role. So, beneath the epithalamus, we have the thalamus. What is the thalamus? The thalamus is a mass of nuclei. We defined nuclei before. Whoops, there it goes. What happened? thalamus. Mass of nuclei. And somebody said they're about 25, plus or minus. We're not going to give you those, just let you know that they exist. All sensory impulses that are going to your cerebral cortex first go through the thalamus before they go to the cortex, with one exception. All right? So all sensory input goes through thalamus before going to cerebral cortex. But there's one sensation that does not. What is that? Olfaction, right. Except that's a course in itself. Why doesn't olfaction? So, for example, just making it easy, we're going to bring light to the retina, to the thalamus, to the visual cortex. And one can do this with all. But you'll find out that the auditory pathway is much more complex, so it's not just from the main receptor to the thalamus to the cortex, so I picked light instead. All right, so that gives you a general idea of the thalamus. What does it look like? They're considered to be egg-shaped. You have the two thalami, a right and a left. And the third ventricle then will be between them, with the hypothalamus below, and then go on down to your pituitary here. So these would be thalamus, thalamus. And the third ventricle will be between them here. And this will be hypothalamus in this slide, in this section. And here's pituitary. Just to give you, this would be a coronal view. All right. In sum, there's a connection between the two thalami. Does anybody know what it's called? The massa intermedia. Massa intermedia. It's missing in 30% of males. And we tested that once in class when we had 20 brains. We knew what sex they were, and exactly 30% were missing it. Males, as you know, if we could go into sex differences in the brain, whole subject, are more lateralized. That means many times they have one side that's much stronger, the right side stronger than the left. And perhaps those people who have this don't have that differentiation, but when it's missing, they have it strongly. I don't know. Now, the structure beneath the thalamus is the hypothalamus. And this, many of us, when we were starting in the field, thought was the most exciting structure in the brain. How much does it weigh? Your hypothalamus. Hypothalamus weighs only 4 grams. How much does a large grape weigh? 4 grams. Gives you the size of it. I know it depends on the size of the grape, but still, what are the functions of this important structure? Why did it attract so many of us when I did my PhD on it? Well, number one, it's a control center for your autonomic nervous system. Control center for the ANS, which we've introduced. It regulates your body temperature. Regulates body temperature. How many have an average body temperature of 98.6? How many don't? How many know what your average temperature is? But how in the world you put all these different kinds of fuels in there, and this machine can maintain a body temperature because you have a hypothalamus. You destroy this nucleus in the hypothalamus. The hypothalamus, again, is a mass of nuclei, just like the thalamus, but only different functions. If we destroy the nucleus for temperature, temperature will climb to 106 and death. Then we have anterior pituitary releasing hormones. This is our pituitary down here. So hypothalamus can send down releasing hormones. Anterior pituitary releasing hormones. It regulates thirst. You finish class, you run out and get a drink of water. It regulates your appetite. Tells you when you want to eat. It affects emotions. Just think. The computation that has to go on to a little structure that's that big. We're still going. It affects mating behavior. It's actually different in homosexual males than heterosexual males in the one area dealing with mating behavior. So 7 affects mating behavior. Then it affects your sleep mechanism. Where are we? 9. Memory. And it produces your posterior pituitary hormones. Produces, we'll put it, produces ADH, which is anti-diuretic hormone to be stored in the posterior pituitary. That was my thesis, to see if it was active in the hypothalamus as well as in the pituitary, and it was. But look at that. Isn't that amazing? You know how you tell somebody where your hypothalamus is? That's exactly it. She knows exactly. Above your nose, above your ear, where the 2 points meet. Right. Because most people have never heard of a hypothalamus. But look at what it does for you. Phenomenal. So let's then go on to our telencephalon. Can I take this off? So we're way up at the top. Telencephalon. The telencephalon then is going to account for 85% of your brain. I mean, it's the big most. And will consist then of the basal ganglia and the cerebral hemispheres. These are the massive hemispheres. So we've worked our way all the way up. When we're drawing embryologically, here would be our lamina terminalis from our neural tube, but then we pouch out like this to start to form our cerebral hemispheres and basal ganglia. This was our lamina terminalis. So that landmark is used a great deal in talking about forebrain relationships. So let's look at the embryo, at the reason why it's called a basal ganglia. Let's look at our hemispheres. And this will be in the embryo. And we'll have the clear till we get to the base, and then we'll come in. So here, in the base of the hemispheres, we will have these masses of cells that are the precursors of your adult basal ganglia. Basal ganglia. And these, of course, are your cerebral hemispheres. And so what will we name or number the ventricles in the cerebral hemispheres? Way back at this level, 1 and 2, right. As we get to the adult, we call them the lateral ventricles, 1 and 2. Will become lateral ventricles. And those who are going to be reading MRIs and PET scans, you'll have to know your ventricles backwards and forwards because they're affected. If you've got a tumor, they get smaller. If you get older, they get larger as the cortex decreases. So ventricles in your medical profession are very important to know. But what do they look like now in the adult brain? Let's take a section through an adult brain. So we'll take our hemispheres again. And we know we have the thalamus in here. Put in a thalamus. And we have a hypothalamus beneath it in here, coming on down just to get our orientation. What's the ventricle between the thalami? Third, right? Very important. Third ventricle. Hypothalamus has the same third ventricle because they're both right together. So it just lets you know what we have in our midline area at this level. But now we're going to put in the basal ganglia. They take on a very different shape in the adult. And we're going to put in some lateral ventricle here. So this will be a lateral ventricle at this level. And we'll put in some more basal ganglia here, inferior to the lateral wall of the lateral ventricle. We've got the same duplicated on the opposite side. We need one more structure. It will be in here. So we're ready to label these now. Basic parts of the basal ganglia. So parts of basal ganglia. Let's take number 1. 1 is the caudate nucleus. Caudate nucleus. We'll show pictures of these. Number 2, we're going to take the lateral aspect of this part. And this is called the putamen. Putamen. And the medial part here has divisions, but we're just going to show it as a whole for introductory purposes. It's called the globus pallidus. These will be second nature to you after you start studying brains. What does that mean? Pale globe. So you'll be able to pick it out because it stains much lighter than the putamen or the caudate, for example. And our 4th one here, I should have had a little bigger thalamus. So it's called the subthalamic nucleus. Subthalamic nucleus. So 1, 2, 3, 4 equal our basal ganglia. Very large, very important part of your forebrain, modifying motor behavior. You get different symptoms from every one of these. If you knock out your subthalamic nucleus, I've only seen one example in my life of subthalamic, and he goes like this. He was an engineer. He could talk to you, he was fine, but he had no control over these flailing limbs. Just amazing. So you better be thankful that your subthalamic nuclei are in order, because I don't see anybody going through such gyrations. Boy, it was amazing, and fatiguing too. All right, so just to let you know the importance of these when they work, you just take them for granted, right? So what else would we want to say about our basal ganglia before we move up? Let's be sure we're catching it. Yes, we want to mention another important structure here. Let's put in the internal capsule. Internal capsule. So we're going to see a pathway, fibers that are surrounding the basal ganglia. These X's make up the internal capsule. And you say, well, is there an external? Yes, but not as much is known about it. But I'll put it in out here. This will be external capsule. But the internal is extremely important because we've put these basal ganglia right in the middle of our hemispheres where we have all this information coming from the thalamus, coming from the spinal cord into the cortex. And it's got to go around these big nuclei. So they call the inner one the internal capsule. So it consists of ascending and descending fibers to and from cerebral cortex. So if I break a blood vessel right in here, we know exactly what fibers will be knocked out coming from my motor behavior, right? These are very well defined within the internal capsule. All right, that gives us one component of our telencephalon. And our next one then are these large cerebral hemispheres, which make you, you, and me, give us our uniqueness to our brains. So cerebral hemispheres. So we're going to divide these into the archicortex, which means the old cortex, and the neocortex. Let's take the archicortex first. It will only have 3 layers in its components. And what are they? They're found in what's known as the hippocampal complex. Hippocampal complex. What does that consist of? It consists of the hippocampus, and the dentate gyrus. Dentate gyrus. We've used this term before when we said that there were neurogenesis, or new nerve cells can be formed in the adult in the dentate gyrus because it had granule cells with small, short axons. So where is this hippocampal complex? Let's put it in and then say a word about its function. So let's take a medial view of the hemispheres. Just to put in some landmarks. We'll see when we cut through the 2 hemispheres, we'll have a big mass of fibers that we've cut through that are connecting the 2 hemispheres. Who can tell me what they're called? The corpus callosum, right. So this is the corpus callosum. For those who haven't studied neuro before, we'll look at them a different way, and we'll see that we've got these fibers that are connecting back and forth to the hemispheres. Coming around and going here. So it's crossing like this. But what we've done, we've cut in the middle. So we've got them crossing and looking at them from a medial view. So that's the corpus callosum. Those are fibers connecting both hemispheres. All right, those for a landmark. Whenever you see those, you know it's a medial view. To pick up the hippocampal complex, we're going to go into this lobe. What is this lobe? Temporal, good for you. Temporal lobe, just, beneath your temporal bone. You can figure these out. This would be which lobe up here? Frontal, sure. So we want to go deep into the lobe because it's not going to be on the surface. So deep within this lobe, we'll find this structure that looks like a seahorse. And to the early anatomists, and this is the hippocampal complex in here. When you study neural, you'll follow all of its pathways, but we're just identifying it here. This is hippocampal complex. And I'd have to peel it apart so you could see the dentate gyrus is right. Why is it called dentate? What does dentate mean? Teeth. You look at it and it's just got all these little rows like this, and that's the dentate gyrus. So it's easy to identify when you have a brain and can peel back the hippocampus. What does the hippocampus do for you? Hopefully it's working in your brains at this moment. What's it doing? What kind of memory? There are all sorts. Pardon? Short-term memory. Right, thank you. So this complex deals with short-term memory processing. Terribly important. And what's another function for the hippocampus? Visual spatial acuity. Visual spatial acuity. So when's it important? Let's say you're going on a trip for 3 weeks. You park your car in the parking lot down in the Oakland airport, and you've got to remember where you parked it. So as you leave it, you turn around, reinforce your visual spatial acuity so you know exactly where it is, so when you're coming from the other direction, you'll recognize where it is. And you need your recent memory to put it into long-term memory. So hippocampus, just a small example. It's very much affected with Alzheimer's. I saw in University of Iowa, Damasio said, "You want to see a defunct hippocampus?" And I said, "Yes." And he said he just has one in from an Alzheimer patient. So you looked under the microscope and the cells were all disoriented and decreasing for recent memory. So you need to work on your recent memory for a lifetime, because otherwise you lose it. All right, so this is the archicortex. And as we said, it's 3-layered. It's not as complex. It's older. But we go up to our neocortex, which is the most highly evolved mass on this earth, and most recently designed. It's the last to develop embryologically and the last to develop phylogenetically. So we're up to our cerebral hemispheres. Let's look at a lateral view again. And as we showed you previously that it's thrown into folds, how big did I say it would be if it weren't folded? Oh, not the cerebral cortex. No, that would really be interesting, because to fold that as big as a cortex, we'd have heads as big as this center part of our auditorium. So to have it, it's as big as 2.5 feet square. And we always ask, why can't you have a head that's 2.5 feet square? Pardon? Well, I think something more important than that. Pardon? Why can't you have a head 2.5 feet square? Why does it have to fold as it evolves? Childbirth, of course. I mean, you imagine any woman who's given birth and knows 2.5 feet square, that's not going to work. So it's highly folded, and it's folded so that you have these folds. And the top of the fold is called a gyrus. And the indentation between the folds, these are gyri, would be sulci, or a sulcus. Some people like hills and valleys. The hills are the gyri. Anyway, just to remember them, because when you have Alzheimer's, the sulci are very much pronounced because the gyri have been losing their cells. So they're important to know. And when we look at the lateral aspect of our brain over here, we have a central sulcus. You have a lateral fissure. Lateral fissure. So, what lobe is this? What lobe is this? What lobe is this? What lobe is it? Temporal, right. So, we've learned that the occipital deals with vision, all sorts of modifications. Part of the temporal, just a little part up here, deals with hearing. The parietal deals with sensory, general sensory, pain, touch pressure. We'll just put sensory. Lots of other functions, but just to give it a name. And the part of the frontal, what's this first part of the frontal deal with? Motor, right? But it's refined motor. And then we have what's called supplementary motor. We'll just give you one other that you've already had. What is here? Broca's area, good for you. What artery is supplying Broca's area? Middle cerebral, right. Don't forget it. You heard the fellows at 131 using that the other day. So this is Broca's area. And what does Broca's area do for you? Motor speech. I wouldn't be able to talk if I had broken my middle cerebral blood vessel. Now, the area that is uniquely human and has advanced furthest in the brain is our prefrontal cortex. So let's say a word about the prefrontal cortex. So, functions of prefrontal cortex. One, planning ahead. How many have thought what they're going to do on Thanksgiving vacation? So you have a prefrontal cortex. Two, sequencing events, knowing how you're going to fit things together to carry out an action. Sequencing events. Initiative. Decide what you're going to do. And then once you decide, is it really wise to do it? Judgment from prefrontal cortex. Working memory. How does working memory differ from others? You hold the memory in your thoughts while you're working with it. I mean, I hold telencephalon. What am I going to bring in next to say? That's working memory, right? So working memory. Oh, lots more, lots more. Let me just pull up a couple just so that you get the dynamics of them. Oh, that's fine. We'll do with those. Now, what game of cards uses all of these functions? Bridge. Yes. That's why we use bridge, to see if we could stimulate the prefrontal cortex, because we found in an immune deficient animal, he was bilaterally deficient in his dorsal lateral frontal cortex. We transplanted the thymus, came back again. We knew we had an area of the cerebral cortex that controls the immune system. So whoops, we've got slides. I didn't see the lights. Did you see the lights? Boy, I was involved. Sorry. Here we go. Can we go fast? Because I've got lots of good slides for you. I'm sorry. First slide, please. No show. Too bad. We've got it. All right. What part of the brain stem are we? Hindbrain, midbrain, forebrain? Midbrain, sure. Here's your aqueduct. Here are your colliculi. Here is the cerebral peduncles. And this is the substantia nigra. You see, it's a major part of that midbrain. In the next one, and our cerebral hemispheres thrown into folds. What area is this? Broca's area. Good for you. What area is this? Hearing, right? What area is this? Prefrontal cortex. Say it. What area is this? Occipital, right. This area? Parietal. Great. Next one. And now it shows what the ventricles look like inside. This is our fourth ventricle back here in the hindbrain. Then we have the aqueduct, this thin one. You can see what happens if a tumor gets against that. So that's really dangerous there to have it such a narrow tube. But then we come up, this is third ventricle. And these are the lateral ventricles as they get stretched out. This would be in the temporal lobe. This will be back in the occipital lobe. And this one will be up in the frontal lobe. So you get all different, but you learn to know those ventricles. Landmarks. Next one. And this shows our brainstem. This was fourth ventricle. Our cerebellum was over here. We cut it off, but we wanted to see the thalamus now. This was all midbrain here. Here we've come up into the diencephalon. The epithalamus would be on the surface in the midline here. We don't see the pineal here on this slide. But then the basal ganglia would be out here with the whole hemispheres surrounding the whole thing. In the next one, and here it shows the basal ganglia. You have to look sharp. Here's the head of the caudate. Here's the putamen, pale globe, thalamus, third ventricle. And you could see this line that divides here. That's that big internal capsule with fibers coming from the motor cortex coming through there. You break a blood vessel, you're paralyzed on the opposite side, because they're all condensed in the internal capsule because of these masses of nuclei that sit here. In the next one, and this shows the fibers as they have to be condensed. So this would be internal capsule from a different point of view, because the nuclei would be in here, coming down very narrow as they come down your brainstem. In the next one, and this shows the dentate gyrus forming, the hippocampus, just to let you see how they're related. In the next one, and it shows step by step how it curls up so that you end up with the dentate gyrus and the hippocampus right together in the hippocampal complex within your temporal lobe. In the next one, and this shows what it really looks like. Here's your hippocampus here, and see the little dentates here? So this is where your recent memory is processed and sent on then to long-term memory. And an article I just read doesn't put long-term memory in the frontal lobe. I sort of have doubts about that, but it did not put it there. It put it in parietal, occipital, and temporal. In the next one, and then this shows that you'll eventually learn the names of all these gyri and what they do. In the next one, and I put this one in. What is this? How many have been to Rome? A few of you. Where is this in Rome? Sistine Chapel, the roof of the Sistine Chapel. Here's God, and here's man. But what's the shape of the shroud that's around God? Did Michelangelo have fun up there at the top of that chapel? Think about it. All right, that's it.